I'm audible, and I would like to start by thanking the organizers, Joanna, and others for the invitation. I'm quite excited and nervous. This is my first ENC talk, and following on Jeff, I'm going to talk about a relatively large small molecule and our work on applying fluorine NMR to larger biomolecular systems. The application of fluorine NMR has been pioneered by several to study biomolecular systems, including the work of Scott Prosser, Kurt Withrick, Angela Gronenborn, and of late, William Pomeranz with the protein observed fluorine NMR. On the solid state side with Mei Hong and Tatiana, and of late also, as well as Gottfried Otting. Now, in fact, the first experiments that incorporated fluorinated amino acids into proteins could be traced back all the way to 1961, where they fed some poor rabbits with 4-fluorophenylalanine, and then they were looking at the proteins expressed by these rabbits, and they found that the proteins were pretty much the same as the non-fluorinated, at least in the case that they have, the 3 proteins that they have looked at. Now, at the risk of preaching to the choir, fluorine NMR has some very attractive features, including the natural abundance, the relatively high gyromagnetic ratio, the bioorthogonality that Lucia mentioned, and to me, the most interesting, it's a sensitivity to its electronic environment that results in a huge chemical shift range for fluorine. But one of the ugly things in fluorine NMR that limits its use to high molecular weight system, it's chemical shift anisotropy that looks like a football in America compared to a soccer ball in Europe, which will be rather round. Now, to put this problem of this CSA of fluorine into perspective, let's take a toy molecule. This toy molecule is a phenylalanine that has a tyrosine that has a fluorine that's in blue over here. It's attached to an amide, and we have a methyl group just to look at some of the key players that we normally observe in protein. And here, thanks to spinach, you see the CSA of these individual atoms. And what looks like the Death Star in pink, that's the CSA of fluorine. Now, this drastically limits the application of fluorine NMR to high molecular weight systems. Now, and as we all know that the CSA scales quadratically with the field strength. And here is a plot of the transverse relaxation rates as a function of magnetic field strength for a protein that's MVP. And you can see the main culprits over here, the carbonyl and the aromatic have a really bad CSA. But when you put that in context with fluorine, they look like little angels. So that's how bad the CSA of fluorine is. So at the time, we were working on methods to leverage the aromatic ThOC that is developed by Pervushin and Withrick for a carbon-hydrogen system. And this was normally limited by the large CC coupling, and that limits your resolution in the carbon dimension to about 60 Hertz. We're trying to use different labeling techniques to see if we can get very high resolution, especially with pyruvate, where we can leverage the aromatic hydrogen carbon ThOC. At this point, we started thinking about, is there a way that we can combat the 2 evils in relaxation, the CSA and the dipole-dipole relaxation for fluorine NMR? Now, here is a spectrum of, it's a carbon spectrum of GB1 labeled with tyrosine. As you can see, this tyrosine has 2 carbons, one attached to the fluorine, 2 C13 carbons, one attached to the fluorine and the other attached to the hydrogen. And what you see is 3 pairs of peaks, and GB1 has 3 pairs of peaks. And here you can see the carbon is split by its coupling to the fluorine, and the coupling is about 240 Hertz. And you can immediately see one set of peaks extremely sharp and the other set of peaks that are broader, which indicates that there is a favorable interference between the 2 relaxation mechanism. And if we choose the ThOC component, we can get some benefits. Now, first, let's start with the question, how do we synthesize such an amino acid, which is labeled C13 and attached to a fluorine? This is a simple 1-step synthesis pioneered by Scott Prosser, and we did this with the help of Vlado Gelev. So you can get this tryptophan commercially available, and you can just, in a 1-step reaction, you can use a select for reagent That'll either put fluorine at 1 or 2 positions, and depending on the stoichiometry and time, you can drive this to create a single fluorinated tyrosine. Now, how do you incorporate this into a protein? It's again rather simple. You do exactly the same way that you do as you grow a protein in a minimal media. Just before induction, you add this terrible reagent called glyphosate, and glyphosate inhibits the shikimate pathway, essentially stopping the synthesis of all aromatic amino acids. At that point, you need to supplement them with aromatic amino acids for the culture to continue to grow. You supplement them with fluorinated tyrosines, and then the tyrosines get incorporated into your protein. You substitute this fluorinated phenylalanine, that'll get incorporated into your protein. So now, here are some of the theoretical calculations for the transverse relaxation rate for a protein with a molecular ThOC of about 25 nanoseconds. And here you can see the decoupled component in blue and the ThOC component in red as a function of the magnetic field strength. So here I compare on the left the carbon that's attached to a fluorine, and on the right the carbon that's attached to the hydrogen. Now we can see that the carbon that is attached to the fluorine, the ThOC relaxation rate is extremely low. And one thing I want to keep you in mind is that even for the decoupled component, the relaxation rate for a carbon that is attached to a fluorine is actually small. We'll come back to this relatively soon. But as in the fluorine, you don't see that much of an effect, that much of a ThOC effect. And what we're going to use is that fluorine relaxes faster due to its CSA. But what we're going to do is that we're going to use the carbon that's attached to the fluorine to disperse this into a second dimension. That's the idea. So here is the ThOC effect as a function of field strength. Here you can see the pair of doublets. And if you're wondering what this small doublet of about 1.7 hertz, that's just the coupling between this carbon and the carbon that is meta to it. And I've compared that with the ThOC effect for the CH. Immediately you can see that the CH is rather broad compared to the CF. And the other thing you'll notice that the ThOC component changes sides. The carbon fluorine ThOC component is on the right-hand side, whereas the carbon hydrogen ThOC component is on the left-hand side. And I was wondering why this is. And when Ilya told me that is because the carbon fluorine J coupling is negative. And that's the first day I even know that J couplings can be negative. So now this ThOC effect not only exists for tyrosine, it exists for pretty much every substitution that you can think about in aromatic amino acids, in phenylalanine, tryptophan, and tyrosine. So here I show you the relaxation rates of the ThOC component for different substitutions, and with the decoupled relaxation rate in blue and the ThOC component in red. And you can see for some of them, it pretty much goes to 0. Now we have a ThOC effect, and we need to now get a two-dimensional experiment to select this ThOC component. And if you read Art Palmer's book and you design this experiment, we think that we want to start with the high gamma nucleus and end with the high gamma nucleus to get the best sensitivity. So we designed a very simple pulse sequence. It's an ST2PT, which starts with the fluorine to carbon, and then you do a ThOC selection, and then you come back to fluorine and detect on fluorine. Now here are 3 different spectra, and what you see on your left is a decoupled spectra, and what you see on the middle is a coupled spectra that should have 4 peaks, 1, 2, 3, 4. But you can see the ThOC component is now we move to the protein MVP. The ThOC component is rather strong, whereas the anti-ThOC component is weak. And now we can do a ThOC selection by the pulse sequence that I showed you to get the ThOC component. Though we are able to get very sharp lines, we are not very pleased because we couldn't see all the resonances that we expected in MVP. Then we went back to spinach and opened it up and looked at the transverse relaxation rate of the different coherences that you would observe in the ThOC. And then when we saw that whenever fluorine is in transverse, it doesn't matter if it's a simple FY or if it's an antiphase magnetization, the CSA just kills it. So you can see that the relaxation rate, and what you see in pink is the contribution of the CSA to that. So we went to a different scheme and decided to keep the fluorine transverse for as little as a time as possible. So we went to an out and stay type pulse sequence, We can either start with fluorine and end with carbon, or you can start with carbon and end with fluorine. This way, you keep the fluorine transfers for a very minimal amount of time. Now, here are the spectra that you can see here. Here's the out and back ThOC, and here's the out and state ThOC. And we prefer the one where we detect carbon because the resolution is in the carbon dimension, and we want to put that in the direct dimension. And here's again a key aspect over here. What I show you is the fluorine spectrum and the carbon spectrum. You can see that both of them are not able to resolve all the peaks, but if you record a 2D experiment, you can get fantastic resolution. Now, this particular tyrosine that we synthesized has a C13 in two places. One of them is attached to a fluorine, and the other is attached to a hydrogen. So you can compare the aromatic carbon hydrogen ThOC and the carbon fluorine ThOC on the same sample. Now, here you see on MBP at about 25 degrees, the CH ThOC and the CF ThOC. And just to mimic a larger protein, we drop the temperature to about 10 degrees. And you can see the CH ThOC is pretty much goes to the noise, where we get beautiful peaks on the CF ThOC. We can now apply this to very large systems, including the 180 kilodalton proteasome, and you can start to see the aromatic signatures of the proteasome. Now, to our knowledge, this CF resonance is the slowest relaxing resonance in bio-NMR today, compared to even the nitrogen component at the appropriate field strength. So these are compared at the appropriate field strengths where they'll be at the maximum. Why is that? Why is the CF such a good thing that nature has provided? And for that, we go back to spinach again. Here, you remember the same amino acid that we talked about, and pay attention to this particular carbon and the CSA of that particular carbon. Now I'm going to play look at the carbon chemical shift anisotropy over here. They're all pretty big. But once you attach that to a fluorine, now if you look at this particular carbon over here, it almost becomes spherical. And just pay attention to that carbon, it becomes almost spherical. So just by attaching the fluorine, you change the carbon CSA so much that even without the ThOC, your carbon relaxes slower. But when you add the ThOC, it relaxes very, very slow. On an average, if you take the square term of the CSA, that's how it appears in the relaxation equation, you get a factor of 2. Again, these are calculations that were done by Ilya. Now, the amino acid that we first started off with was tyrosine, and we put the fluorine at the 3rd position. This has one problem because we all know the aromatics ring flip, but now the ring flip will no longer be symmetric, and there'll be preferred positions depending upon where the ring flips. And that's one of the problems. And the second problem is that the ThOC is only enjoyed by the carbon that is attached to the fluorine, but not by the fluorine. So we thought we'll mitigate these two problems by a new strategy. And that strategy was to make a phenylalanine with the fluorine at the 4 positions. And now you don't need to worry about ring flips because it's still going to be symmetric when the ring flips. Now, again, with the help of Lather Geller, we synthesized a phenylalanine that looks like this, where we have a fluorine over there. And then we deuterated these 2 positions. Why? Because we can. And 2, you'll understand why I'm going into in the next couple of slides. Now, here is a beautiful spectrum again of, now we switch to a sample human carbonic anhydrase. And here's a beautiful spectrum of the phenylalanine, the ThOC, at both fluorine detected and carbon detected. At 25 degrees and 5 degrees, we still see very beautiful peaks. But in order for us to record such an experiment, you need a probe that can pulse on fluorine and carbon. And the other thing is that the fluorine still has the problem of the CSA, even though we can disperse it in the 2nd dimension. Now, what we asked the question was, can we get the best of both worlds? Can we take this hydrogen that is at a meta position to this particular carbon, which is rather isotropic, and then correlate to the ThOC component of that carbon, leveraging what the fluorine can give you? So to this, we decided to code a very simple pulse sequence. It's a head core. Essentially, you transfer magnetization from this hydrogen to this carbon. And then during this process, you encode the chemical shift of this hydrogen. Then all you need to do is refocus this carbon with respect to this hydrogen and detect the carbon. While the refocusing is happening, I can introduce a 180-degree pulse on fluorine to create an in-phase, anti-phase signal of carbon with respect to fluorine, and you can choose the ThOC component with respect to the fluorine. So that is the idea. The idea is to correlate this hydrogen with the ThOC component of the carbon attached to the fluorine. So I encoded the pulse sequence, we did the experiment, and I didn't see any resonances for the anti-ThOC part. And I've spent about 3 to 4 hours seeing what is wrong with the pulse sequence before I realized that the anti-ThOC part has already relaxed out. So you don't even need the IPAP for the larger systems. And just to make sure that this is the case, I took a very simple HSQC and I appended a refocus delay at the end of it. This is an HSQC going from this hydrogen to this carbon, and I simply appended a delay that is refocused during the encoding time period. Now here you see with the delay set to 0, you can see 2 peaks, the ThOC and the anti-ThOC. When I set the delay to 8 milliseconds, the anti-ThOC seems to vanish out. When I set the delay to 12 milliseconds, I completely get the ThOC component. So for larger proteins, this becomes a very simple experiment. You just a simple head core experiment, you go from hydrogen to carbon, refocus it, and then you have your spectra. So now here you can see that I'm correlating this hydrogen, which is about a 6.5 ppm, with this carbon that's attached to the fluorine, which is now shifted to about 160 ppm. You can see at 5 degrees, you get beautiful spectra. And the important thing of this is you don't even need an F19 probe. You can take a standard TCI probe, run this experiment. Now here comes a more philosophical question. I showed you 2 ways of separating them. Here I show you the carbon-hydrogen correlation. Here I show you the carbon-fluorine correlation. And here are the comparative line widths. You get very similar line widths on the carbon, whereas on the hydrogen, the line widths are extremely narrow, whereas in the fluorine, the line widths are pretty broad. But if you look at the resolution, the fluorine is spread over 10 ppm, whereas the hydrogen is spread over 2 ppm. Now, this almost becomes a philosophical discussion as to what is resolution. Is it the line width, or is it the ability to see each one of your peaks? And there, you're left with your own cup of tea. Now, this fluorine carbon ThOC effect not only works in proteins, it also works in nucleic acid. Actually, it works much better in nucleic acids. So here again, theoretical calculations for the substitution of fluorine in different nucleotides. And you can see the ThOC effect causes very slow relaxation rate for most of them. So when we were preparing our first manuscript, we didn't have a way to get to a C13 F19 labeled nucleotide. So we just ordered an F19 labeled nucleotide from IDT and used a natural abundance to show that the ThOC effect does exist in DNA in this particular case. Here you can see the 4 component. One component is clearly much more sharper. Now, after we published this, there's some very interesting work from the group of Christoph Kreutz, who has synthesized these C13 F19 based nucleotides, and using a DNA synthesizer, able to incorporate them into selective places in RNA and get very beautiful spectra. And here's the work from Owen and Kwaku, where they use runoff transcription to incorporate these C13 F19 based nucleotides and get, again, fantastic spectra. Now, in the last 1 minute, what I'll do is that I'll talk about a possibility of, okay, now we can assign these, but we can incorporate these into proteins. How do we assign them to make them very useful? So one way we can do them is a sure way is mutation. We can mutate each one of, if you have a tyrosine, you can mutate to the phenylalanine, make 10 different samples, and then you can assign them, sure shot. But for crying out loud, this is ENC, we need to have some 100 spins. So one of the other ways that people have tried, and we also tried, is using a ThOC experiment. If you have the proton assignments, you can do a heteronuclear ThOC between hydrogen and 19F. That is definitely not a ThOC. ThOC is not very sensitive. But the problem with this experiment is that it's least sensitive. It's not very sensitive. The other type we tried is using a cross-saturation transfer type experiments. In the case where your fluorine resonances are far apart, like you have it in a GB1, what you can do is you can saturate selectively one of the fluorine resonance and then record an HSQC on and off resonance. Now, what you'll get is that you'll get these beautiful peaks. Now you can see that when you saturate the resonance corresponding to tyrosine 3, these peaks light up. This is essentially what I'm showing you is a different spectrum with on and off resonance saturation. And then when you saturate a different, then you get a different peak, and that helps you assign them. But the problem is that it'll work for small proteins. For larger proteins, you're going to have signal overlap in the fluorine channel, in the fluorine spectrum, and you'll not be able to see this. Now, for this, we took a page out of Jim Prestegard's playbook, and then we started to use RDCs to get the assignment. What you should note is that the ThOC is only enjoyed by the carbon, yet you get two very sharp lines along the fluorine. Now you can use them to measure dipolar couplings. Now, if you, for example, in this case, with the protein human carbonic anhydrase, you can use the NH assignments to get the alignment tensor. Then you can go to spinach and then ask spinach to calculate, knowing the structure and the alignment tensor, what would be the dipolar couplings of the different aromatic amino acids. You measure the dipolar coupling from your CF and you just pick your peaks up. You can pretty much assign almost all the resonances using this particular method. So there are different ways to do the assignment without doing the painful mutation. So in conclusion, I think I'm hoping to convince you that the CF ThOC provides you an opportunity to probe structure, dynamics, and interaction of large proteins and nucleic acids. You have good assignment strategies in progress. This really does not depend on deuteration. And as shown by Lucia, you can also make these proteins in insect cells and other cells. And RDC measurements can not only help you in assignments, but also can give you critical information about dynamics as well. With that, I would like to thank the people who did the work. The work was pioneered by Andras, Abhinav, and Thibault. And we wouldn't have done this. Even the initial ThOC effect came from a remnant of Ilya's thesis, which Ilya didn't even recognize there was a ThOC effect there. But it was there in his thesis, the synthesis of these amino acids by Vlado. And for a long time, we didn't have a fluorine probe, and I couldn't have done any of these experiments without the help of Bruker. With that, I thank you for your attention.
That's beautiful work, Ari. I guess we'll start with questions. Over here. Go ahead.
Hi, Harry. Great talk. I noticed that a lot of the fluorine probes that you're using have only a single fluorine. Have you looked at other types of probes, like maybe something with like a CF3 or maybe multiple fluorines on the same amino acid? And do you think you would still notice the same effect, ThOC effect, with different amounts of fluorine in your probe?
The question of CF3 has been, we thought about it, and actually there's a beautiful paper by Brian Sykes. So the CF3 ThOC effect is a different mechanism. It's not a CSA dipole mechanism, it's a dipole-dipole interference mechanism. And for CF3, that ThOC effect does not exist. So Brian has published a paper, but I'm not really sure if the same thing would exist for a CFD2 or CF2D. That's something I think is an open question. Now, the ThOC effect when it comes to aromatic amino acid is strictly a single bond dependent, unless there is a lot of electronic difference in the environment. We didn't try to go to multiple fluorine because even putting one fluorine in there is going to cause perturbations to the structure. And we don't want to put a lot more fluorine and cause more perturbations because we're looking at one probe. That's why we didn't think about putting multiple fluorines in there.
Thank you.
That's a good point, because if you're doing enzyme assays and there's probably going to be a fluorine effect on your activity, you got to make sure that's not terrible, right? Okay. Mutual politeness.
I'm a little intrigued by the spinach calculations. How much tensor information do you need to give it? In other words, is it actually getting the tensor by first principles calculation, or do you have to provide that and then you can then do these other computations?
Ilya can answer. I think it was using Gaussian, the tensors are predicted, and we also have an experimental validation with Tantiano's group. Okay. Ilya says that we don't have to measure tensors anymore, but the tensors calculation was right on spot. Ilya, do you want to expand on that?
Yeah, Harry is right. It's just a straightforward DFT, and then spinach imports Gaussian logs directly.
Okay. I mean, I believe that. I actually just wanted to hear you say it, Ilya.
So thanks for a great talk. Just one thing I might have missed. I just wanted to clarify. So you see increases in the carbon T1 with the T1, right? With the fluorine addition, like for all relaxation, or
It's the T2 of the carbon that goes up, not the
Oh.
T1. Yeah.
okay. But the T1 is that
The T1 is pretty much the same for the carbon and the fluorine T1 is also about very similar to the hydrogen. And that was measured by us. And there's a beautiful paper by Angela Cronenboom. They also have measured the T1 of the fluorine attached to amino acids, aromatic amino acids.
I see. Okay.
Yeah. And that's why when you do a fluorine start, carbon end, you can actually go with the T1 of the fluorine, which is smaller than the carbon. So you can pulse faster.
If you have, I mean, one application for this obviously could be, if you are screening small molecules for binding at a site with fluorine at it, that would seem, it would be very sensitive, I would think. But then there's the issue of interpreting what those chemical shift changes would mean for fluorine. If you, I don't know if that has entered your work thus far, but interpreting what F19 chemical shift perturbations mean, that could be useful.
We haven't gone in that direction.
Oops, sorry.
Rasmus?
Yeah, beautiful talk. I just have a question to a very small detail, which I didn't really understand. So in this first spectra that we showed, where you just copied Prevushin's like normal ThOC sequence and Vitris, you basically saw this 2J coupling between the two carbons, and then obviously for the other phenyl.
You mean this spectra?
Yeah, exactly. And so this doublet, it didn't show up anymore afterwards, right? In your direct detection.
Because if you go further into the thing, because here it's in the direct dimension, so we can collect as many signals. But when you do the 2D, we don't collect as many signals, and we also aperize the data differently.
I see.
Yeah, so
it's
this will
basically-
definitely show up in the carbon direct dimension
Okay.
as well.
No specific tricks.
Yeah.
No encoding.
And the 2D spectra that I showed you after that are on MVP. They're a lot broader than GB1.
I see.
So if you take GB1, you'll definitely see these doublets.
That explains
Yeah.
it. Okay, thanks.
All right, if there are no further questions, let's thank Hari again for an inspiring talk. So our next presentation will be from Jim Prestigard from University of Georgia, and on a topic that was very timely, the importance of another kingdom of small molecules that, when attached to big ones, have significant effects. So whenever you're ready.
All right, thank you. I think I'm just going to get directly underway here, and I'll put up this picture of this molecule. Many of you probably are tired of seeing this at this point. This, of course, is the spike protein on the outside of SARS-CoV-2, the virus that's responsible for COVID-19. I'm not actually going to talk about this molecule, but it's a nice illustration of the importance of the glycans that exist on the surfaces of glycoproteins. Now, of course, you know, our own proteins in mammalian cells, more than half of those are actually glycosylated and composed of not just amino acids, but have attached sugar residues. And they serve, of course, a variety of purposes. They mediate the interaction of a cell with its environment. They're involved in controlling the lifetime of proteins in your bloodstream. They're involved in the quality control folding mechanisms in the ER and so forth. And for viruses, they are important in what is often called a glycan shield. All right, so this is a picture that was developed by one of my colleagues, Rob Woods. And the glycans are on here are this purple and green fuzz that you see. All right, and the message is that it almost completely covers the surface. And there are only a few, such as the circled area here, regions where the amino acids that are useful in developing antibodies are really exposed. Now, this picture is maybe a little bit deceiving because it's not a picture of just a single glycan on all the sites. It's actually a superposition of snapshots from a long molecular dynamics trajectory, superimposing glycan positions from every nanosecond. All right, and so the question arises, you know, is this accurate? Are the glycans really flopping around like this, or do they actually have preferred positions? And that's really the question that I want to address today and show you some methods and techniques that we've been developing to try to address questions like that. All right, so the molecule I'm actually going to talk about is this one, CCAM. And you see a ribbon diagram. That's just the N-terminal Ig-like domain of this cell membrane attached molecule. It has several such domains extending out. It's a highly glycosylated system. Even this single N-terminal domain has 3 N-glycosylation sites. And these are depicted here in stick diagram form. And over here in terms of this cartoon that is referred to as the symbol for glycan nomenclature, in which N-acetylglucosamine residues are the blue squares and mannose residues are the green circles. This particular piece that we work on has actually been engineered to be almost fully glycosylated on all 3 sites and to contain only this particular form of a glycan. So really a native glycosylation, a very diverse set of different types of glycans. Now, so we're going to study this by NMR. And of course, the fact that to get native glycosylation on these systems, the best way to do it is express the protein in mammalian cells. And mammalian cells present some obstacles in that you can't just feed them very fundamental substrates like glucose and ammonium chloride and label all the amino acids or the glycan. So glycans actually you can, but it's to study the protein itself, you're going to have to actually feed isotopically labeled amino acids, right? And you can do this, of course, to get a uniformly labeled system, but it gets expensive for one thing. And also, there's a problem in that mammalian cells do not tolerate deuteration. And so you can't perdeuterate. And if you label all the sites, of course, your spectra get rather complex and overlapped. So we use a strategy that we refer to as sparse labeling. And that is simply to select 1 or 2 or few types of amino acids for labeling. And in many applications, this is just fine. I mean, you're often presented with a structure that you know, and what you're really looking for are surface interactions. In our case, where are those glycans related to the membrane surface? And for that, you don't need to necessarily label all the sites. So the strategy we use, and what I'm going to show you, we've added dimethyl C13 labeled valine to the medium, and we've also added C1 labeled glucose. Now, the C1 labeled glucose, of course, goes very readily into all of the sugars. All right, so all of the anomeric sites in the N-acetylglucosamines, the mannose, are going to be labeled. C1 glucose also goes through glycolysis, of course, and gets down to pyruvate, and that C1 carbon ends up in the methyl group of pyruvate. And pyruvate, in single enzyme steps, goes out here to acetyl-CoA, and in turn labels all the N-acetylated sugars, the N-acetylglucosamine, And in a single step by an alanine transaminase, it goes into alanine and labels the alanine methyls. All right, so we end up with a system that has labels in all our sugar residues. It has valine methyls and it has alanine methyls. Sparse labeling has some downsides. You don't have a dense enough set to really expect to capitalize on nuclear Overhauser effects very much. And in particular, our glycans, which may be flopping all over the place, don't have very many close contact. So you need longer range probes of this. And the way we do this is by introducing a paramagnetic site, in particular lanthanide iodine, that can produce pseudocontact shifts or paramagnetic relaxation enhancements. And the equations, of course, are given down here. They're clearly geometry dependent. All right, 1 over r cubed and 1 over r to the 6th, which is very desirable, and the range is large, right? So you cover, 30 angstroms, maybe even 40 angstroms in some cases. Certainly enough to look at where the glycans are. However, you need to parameterize this, right? So the pseudocontact shifts, you need to know where that ion is, and you need to evaluate the anisotropic or the rhombic and axial components of the susceptibility tensor. All right, and so the way we're going to do that is actually starting with a known structure of this domain, and we're going to use the effects on the amino acids to evaluate those parameters, and then we're going to come back and look at what happens to the pseudocontact shifts and paramagnetic relaxation enhancements of the glycans. So we started actually with an alpha-fold structure. Our lanthanide is actually bound in a little peptide loop that's been inserted into the expression sequence. This is something developed by Barbara Imperiali's lab many, many years ago, and we've used it on other proteins. The only thing that's different here is that we had a little bit of trouble with this introduction, destabilizing parts of the protein and reducing the affinity for lanthanide ions in some cases. It turns out you can stabilize this by putting a disulfide, pair cysteines in a disulfide bond. All right, and that works in fact very well. It turns out that geometry not only stabilizes the loop, but it's completely compatible with the geometry of anti-parallel beta strands. And so it works very well for a number of sites in this molecule. Let me go on. So what do the data look like? All right. And so this is actually a carbon detected HSQC that we're using here. There's some reasons for that. We could go in during questions if you like. But the black cross peaks here that you see, those are with the diamagnetic lanthanide lutetium added. All right, so no pseudocontact shifts and no relaxation enhancement. And then the red are with terbium, the paramagnetic species, and you see that there are pseudocontact shifts as indicated by the blue arrows. We also have gadolinium, which does relaxation. I'm not going to show that particular data. The alanines here are all of these that are above about 1 part per million in ppms, and all of the valines are up here. So that's what the data look like for the protein labeling. And we're going to go on and try to extract those parameters. But the problem is, of course, we need assignments for those cross peaks. And we don't have the uniform labeling that allows you to walk from one nucleus to another through scalar couplings. All right, so we devised a program that we call Assign SLP that does this assignment for us. It uses a variety of data Initially, it was focused on chemical shifts that come out, of course, directly out of HSQC spectra, nuclear Overhauser effects, residual dipolar couplings, but we've added now pseudo-contact shifts, ThOC data, the PREs and so forth. You can use any combination that you like. It's based on a genetic algorithm search for the best match between predicted assignments, all right, which are done in various ways within the program, and also externally, we download chemical shift predictions from any number of programs that are out there. And it matches those to your experimental data, all right? And this will generate an assignment in the matter of less than a minute, just very quickly. It's presented in terms of heat maps, not like this one. This one is a little different because the genetic algorithms will always find an assignment. You're never quite sure how good the assignment is. All right, so there's a module within this, which we call a validation module, that goes in and does Monte Carlo sampling within error estimates for all the experimental data and reproduces this search. All right, and then the statistics of those results are analyzed to give confidence levels. All right, so the dark blue squares here are actually 95% confidence levels. And there's quite a number of them. Some of them we can check. All right, for example, this one is, that's a pair of valine methyls on, that's valine 44. All right, and for several of the valines, actually, we've done mutational assignments, and we know that one is absolutely correct. For the alanines, there's actually some data from the Gerhard Wagner lab that's done backbone assignments for this molecule, and those go out to the beta carbon on alanines. We don't have proton shifts from that, but we have the carbons, and so we can look at the outliers in our carbon shift and compare to those assignments. And again, all of these dark blue squares correlate exactly with those assignments. So this appears to be really quite a reliable tool. So with that, we get our assignments. We can go back and calculate pseudo-contact shifts. And those are presented here, superimposed on our molecule as a constant pseudo-contact shift surface at 0.1 ppms. Both being upfield in pink or red and downfield in blue. All right, and this is just an expansion. And what you see is if you look at the glycans, this is just an arbitrary conformation of glycans. We expect to see many of those glycans in an upfield shift region, many in a downfield region, many small pseudo-contact shifts that are further out. We could draw spheres further and further out here. And so this is what we predict. And then we go and look at experimental data. This is the N-acetyl methyls for our glucosamines. And you notice we can start with the black and compare lutetium to terbium, and the shifts excuse me, are out there. But you notice one thing, they're all, most of them rather large, and they are all upfield, all right? Nothing downfield. And the same thing happens if we go over and look at the anomerics. You can actually classify and do partial assignment based on the results of the Casper program from the Vidbaum laboratory, and those are sort of labeled there. But again, lutetium versus terbium. We get almost total resolution of these in the terbium case. But again, you'll see the shifts by and large, they're fairly large and they're virtually all upfield. So that snapshot I showed you just doesn't quite make sense. So if you go and look for what are some other conformations, the first thing you do is do a molecular dynamics trajectory. And this is monitoring distance between one of those acetyl methyls and some surface sites on the protein. You see it goes along, all of a sudden there's a transition. to some other state. Actually, it goes from small distances to long. So, it's one of these spread out structures. Is it real? Well, the answer is no, it's not. The problem is, even a microsecond MD is far too short a molecular dynamics trajectory to explore those conformations. So you go over to something like accelerated MD. This is Gaussian accelerated from the McCammon and Meow laboratories. And you see these, even though these jump up to those extended states, the majority are these close contacts, all right? And so if we pick one of those, and we can rescore those accelerated MD trajectories to get most probable clusters and look at representative structures. This is a tool that does that. And here's the superposition with the most probable structure. And you see this glycan has come down along the surface, all right? And now it is mostly upfield or very small chemical shifts, pseudo-contact shifts. And if I actually look at the structure, there's some rationale for this. This is the N-acetyl methyl, and it's snuggled down into this hydrophobic pocket on the surface of the protein. This is a leucine contact. This is a epsilon proton of a histidine contact. If you go back and look at the nosy data on those, you can actually find there are chemical shifts that correlate with those type of contacts. So this is a start. Obviously, there's a lot of work to do here looking at ensembles and more detail of additional structures and proteins, and we hope to do that in the future. So with that, I'll just put up my acknowledgement slide here and let you look at that and let you ask questions at the same time. Thank you.
Thanks, Jim. That was a fascinating talk on a lot we don't know about the functional mobility of those glycans. Questions, please.
Hi, very nice talk. I was wondering, have you looked at residual dipole couplings from these lanthanides? I mean, CH groups should also be suitable for that?
Yes, we have in some other proteins. And we did for this one too, in an early version that had just the valine methyls labeled. And it turns out there's sort of an unanticipated problem. Maybe we should have anticipated. We were trying to be very clever labeling two methyl groups with valine. But it turns out that roughly half the valine actually are not stable. They do flips about the chi angle. And that has just horrendous averaging effects on the RDCs. And so basically we end up not being able to use them. So you certainly can collect. We didn't collect them on the alanines. The alanines should work just fine, but we just haven't got those data yet.
And what about the glycans? Can you see them in the glycans?
In principle, yes. We don't know exactly how much averaging is going on. If there's not a lot of averaging, we certainly should see those for the glycans.
Thanks.
Thanks, Jim. Beautiful. My question's a little bit about the averaging at the end. So you have these MD trajectories, and so you have states. So you have some, or you at least presume you have a population of states. Can you use that, or does that inform in any way the PCS, the average PCS that you see? Because it's what you see is the ensemble average of these trajectories.
Yeah.
And
Yeah,
does that help you look backwards a little bit?
Yeah. That's obviously the next step is to try to do that. You'd like to be able to take the probabilities predicted by correcting the energies in the accelerated MD, but I don't think they're actually accurate enough to, just use those simply because the energies in molecular dynamics, are only good to about a kilocalorie, and that's a huge effect on populations. So I think our strategy is we're going to just take everything that's more than 5% and then adjust populations and see if we get better fits. That would be the strategy.
Okay, thank you.
I'm very intrigued by the use of the carbon detected because do you also choose like what type of a pulse sequence do you choose for the ThOC, the methyl ThOC, and why did you choose the carbon detected?
Yeah, so, I mean, these are fully protonated systems. So the methyl ThOCs are not as good as they are in a fully deuterated protein. The carbon detection on molecules that are sort of in the 20 kilodalton range, we've done as accurate a comparison as we can with a carbon optimized probe at 600 and a proton optimized probe at 600. And we only lose about a factor of 2 in sensitivity. All right, so that's a modest sacrifice. And the fact is that the resolution in the carbon dimension is so much better. Not only, just inherently, but the other thing that happens is it turns out that spending more time on proton transverse brings in chemical exchange effects on protons that are inherently larger than they are for carbon. All right, so we lose more peaks due to phenomena like that as well. So there are a couple reasons for going over to carbon, and presumably that's going to get, those reasons will get stronger as you go to larger molecules.
And also higher fields, I think, is a great thing too, because at higher fields, your proton sensitivity also goes down with the salt, whereas the carbon sensitivity is rather immune. So
yeah, that too. And of course, also with the paramagnetic ions, that the broadening effects are less if we do carbon detect. That's another reason.
Okay, we got. It's time to move on to our next speaker, but let's give Jim thanks again. Okay, our next presenter is Jiaqi Liu from NYU. And it's an intriguing title, certainly in the abstract, about a big problem that has been hiding in the dark, I assume.
Hey, all right. Hello, everyone. I'm Jiaqi from Gershaw Lab at New York University. First, I want to thank ENC for giving me this opportunity. I feel very honored to be here and give and introduce about our work on liquid state phosphorus NMR. So my topic today is the phosphate form spectroscopically dark state assembly in common aqueous solution. As we all know, phosphate and polyphosphates are molecules with simple structure, but they play ubiquitous roles in a myriad of processes, both in chemistry and biology. And what inspired us most to study the phosphate is their potential role in quantum neuroscience. In a recent literature, Quantum Cognition, it was mentioned by Dr. Matthew Fisher that the quantum effects might indeed play some role in human cognition. It was proposed that the phosphorus nuclear spin could serve as the rudimentary qubits, which entangle in the network of poster molecules, calcium phosphate in our brain. And they're able to store information over potentially long time and enable our brain to function then. However, we know this is a controversial hypothesis for now. It still needs a lot of work to prove its correctness. As part of the collaborative team with Dr. Fisher, it motivated us to study the NMR properties of phosphate in aqueous solution in detail. Therefore, we measured a series of phosphorus NMR spectra for orthophosphate solution as a function of temperature. Each spectrum you can see here consisted of a single line shows significant browning with increasing temperature. And to test the veracity and generality of this phenomenon we observed, we repeated these measurements on many other aqueous solutions of phosphate-containing species at different solution conditions. For example, like different concentrations, different pHs, and even different counter-ions. And you can see in this plot, in addition to orthophosphate, we also show the line width measurements for pyrophosphate and ADP as a function of temperature. As we can see, even though the extent of the line browning varies for different phosphate-containing species and solution conditions, The phenomenon of this kind of increasing trend is common among all these phosphate containing species tested here. So therefore, we believe this phenomenon suggests that there is a common underlying molecular mechanism for the solvent-exposed phosphate groups. Actually, the line broadening result we got is very surprising because according to BPP theory, increasing temperature should lead to motional narrowing of NMR resonances for small molecules as their tumbling rate increases. But our results are inconsistent with the expected trends for small molecules, including the ionic species. Therefore, to further explore the nature of the underlying process, we measured the phosphorus NMR spin-spin relaxation R2 and the spin lattice relaxation R1 for the orthophosphate solution with different concentrations at varying temperatures. And in this plot, we show both the line width derived R2 and the CPMG measured R2. Comparing to the line width derived R2, the CPMG measured R2 allow us to assess whether this kind of line broadening is originating from the inhomogeneous broadening. As we can see in this plot, we found that each two of them show the comparable trends of the line broadening, and with slightly higher values of the line width derived R2, it's probably due to some field inhomogeneities and small temperature gradients. So the R2 measurements verify that the phosphate line width is primarily broadened by the dynamic properties of a homogeneous spectral population. And then let's look at the R1 measurements. We measure the R1 for the orthophosphate solution with different concentrations as well. And we also observe some unexpected trends and values. As illustrated in BPP theory, the small molecules tumble in the fast motion regime, and their R1 and R2 are expected to be monotonically decreasing, and R1 is expected to coincide closely to R2. However, we observe that the temperature dependence of R1 for the orthophosphate at higher concentration show a local maximum here. And besides, the R1 values are nearly 2 orders of magnitude smaller than the R2 values for the same sample. And the R1 measurements suggest that the monitored phosphate species are experiencing much slower molecular dynamics than those of the isolated orthophosphate monomers. Therefore, we got some hypotheses to interpret the phenomenon we observed. First, there is an invisible pool of phosphorus spins. We call it dark state because they are spectroscopically invisible. And these phosphate molecules are assembling into larger species with tumbling rate in the slow motion regime at elevated temperature. Besides, the ionic phosphates are in exchange with the spectroscopically invisible species with greater relaxation rates. And higher temperature may facilitate the growth in population and size of the assemblies and maybe accelerate the exchange rate at elevated temperature. So in order to prove the hypothesis, we need some experiments beyond the 1D spectroscopy. In our study, to test whether there is an exchange between the visible species and the spectroscopically dark population, we performed the chemical exchange saturation transfer. In this case, you can also call it DEST for dark state exchange saturation transfer. We achieved the phosphorus test by saturating a selected region in the spectroscopically invisible region of the spectrum, and followed by the detection of the visible signal, which is in exchange with the species below the NMR detection limit. And these experiments were repeated with different saturation frequency across the complete spectral region to allow us scanning the whole spectrum for the potentially exchanging species. The exchange can happen many times during the pulse saturation and transfer the saturation level between the visible and the invisible species so that the enhancement effect for the detected species can be achieved this way. And then we can test the existence of the species at dark state within the scan frequency range. And the plot of the detected intensity versus the saturation frequency is called a Z spectrum, and it's centered around the visible phosphorus NMR signal. Here we show both the simulated and the experimental set data. The simulation was achieved by using the exchange model. The thermodynamic parameters fit the experimental sets very well. And figure B shows both the measured and the simulated Z spectral deep width as a function of temperature for the orthophosphate solution. It can obviously seen that the deep width increase with the increase in temperature, which is pretty much consistent with the line browning trend we observed in our R2 and the line width measurements. And figure C shows the pulse power dependence of sets. In this figure, we can see that in the absence of exchange, one expected the Z spectral deep width to be 2 times of the irradiation pulse power in Hertz. Well, in our case, we observed that the Z spectral line width as a function of irradiation pulse power to be significantly larger than the 2-fold of the irradiation pulse power. Therefore, these results further confirm that there must be significant underlying exchange between the visible and the invisible species. Besides, with temperature increases, the exchange rate becomes faster. After indicating the formation of the phosphate assemblies, we also examined whether their formation is reversible or not to see whether this assembly is thermodynamic or kinetics. If the process is reversible, that means the assembly is thermodynamic, otherwise it's kinetics. So we tested this process by characterizing the phosphate at 298K, 343K, and cooling back down to 298K. The results show that the phosphorus NMR line width, the phosphorus sets, and also R1 and R2 are all reversible. And you can see the numbers on the bars here are showing the ratios of absolute integral of the phosphorus NMR peak. And the significant decrease of the absolute integral at higher temperatures further confirm the greater formation of phosphate clusters in dark state. So our results on reversibility indicate that the phosphate clustering is primarily a thermodynamic process. In addition, there is compelling evidence for assembly formation. The previous measurements didn't provide us the direct observation of the phosphate assembly because of their spectroscopically dark nature. And we hence use the cryo-TEM to determine whether the phosphate assemble into large and persistent enough clusters to be imaged. And you can see in the images, all the solution tested show the evidence of the phosphate assemblies forming at sizes ranging from 20 to 50 nanometers at diameter. And we next use the MD simulation to see whether the monomeric phosphate species can form stable clusters as well. And we simulated 3 different solutions. There are monohydrogen phosphate, the dihydrogen phosphate, and the 1-to-1 mixture of these 2 phosphate species. At 343K, the cluster distribution of the phosphate system is represented as the free fraction Pn of the phosphate ions in a cluster of size n class. And in this plot, we can see that the monohydrogen phosphate shows the strongest tendency to assemble, and followed by the mixed phosphate system, and then the dihydrogen phosphate system. So overall, the MD simulation results also confirm that the orthophosphate cluster formation under comparable conditions as the experimentally proved. Having established the larger phosphate assembly exists in the aqueous solution, we next explore the potential mechanism for the assembly, in particular temperature behavior. And we performed phosphorus ThOC NMR on the orthophosphate solution. And the plot of the hydrodynamic diameter as a function of temperature is converted from the diffusion coefficient plot by using the Stokes-Einstein equation. And we can see here the results show great reversibility and also the significant decrease of the hydrodynamic diameter at higher temperatures. Therefore, we hypothesize that the phosphate ions are experiencing partial dehydration with increasing temperature, and the dehydrated phosphate groups can more readily assemble into dynamic clusters. And then the question is, what is the driving force for the formation of this kind of soluble, non-covalent phosphate clusters at elevated temperature? Considering the Gibbs free energy, we notice that it would occur when delta G is negative. And there is no doubt that with temperature increasing, the enthalpic gain delta H must be positive. So the entropic gain delta S should be positive as well. And you can imagine that during the process of temperature increases, the entropic gain of shedding hydration water becomes more heavily weighted than the entropic decrease of phosphate assembly, hence lowering the Gibbs free energy. And the possible source of the entropic gain is depletion interaction. So the depletion interaction is kind of attractive force resists between the large particles that's suspended in the dilute solution. And in this process, the small particles are preferentially excluded from the vicinity of the large particles and allowing the network interactions that give rise to the formation of the larger clusters. Therefore, the results we got suggest that the phosphate clustering is an entropic process driven by the dehydration of phosphate clusters. To prove this interpretation, there are two ways we employed to amplify the dehydration process. One is adding the hydrophilic molecular crouters, the polyethylene glycol. The other one is adding the salting out salts along the Hofmeister series. As we all know, the PEG has strong affinity to water, so it drives the dehydration. The results here show the overall R2 rates increase with the increasing PEG concentration. And the observation is consistent with the interpretation that PEG facilitate the dehydration of phosphate and subsequent clustering if we consider the line browning as a proxy of the dehydration-induced clustering. Plus, the line with derived R2 in the absence of PEG and in 18% of PEG as a function of viscosity indicate that the line with browning cannot be considered for solely due to the changes in the solution viscosity from the PEG because these two lines are not superimposed. And then let's look at the dehydration interactions modulated by the addition of various salts based on the Hofmeister series. The line with R2, the line with derived R2 show the ammonium causes the greatest line browning and followed by the potassium and then the sodium. Again, if we consider the line browning as a proxy of the dehydration-induced clustering, these results are completely in agreement with the predict trend of Hofmeister series. So perfect. And therefore, these results also add further supports to our hypothesis that the phosphate clustering is an entropic process driven by dehydration of phosphate clusters. In conclusion, in this study, we found an invisible pool of phosphate cluster in aqueous solution, and these phosphate clusters with fast relaxation rates in dark state are in exchange with the ionic phosphates. In addition, we have presented strong evidence that this phosphate clustering is an entropic process driven by dehydration of phosphate clusters. And the more important thing is that our study suggests that this kind of phosphate clustering might be existing in numerous phosphate-containing species at a wide range of solution conditions. Considering the ubiquity of phosphate in the biological studies, we propose that such assemblies should be considered in the interpretation in both in vivo and in vitro experiments. And last, I would like to express my appreciation to my advisor, Dr. Gershow, and our postdoc, Dr. Karanchan in New York University, and all our collaborators in University of California, Santa Barbara. This project cannot be achieved without their hard work and dedication. And also, please allow me to make an advertisement for our group. If you are interested in our projects, please feel free to contact us. We're looking for postdocs. And also, thank you for your attention, and I'm happy to take any questions.
Thanks very much. I remember when that Posner paper came out by Fisher, it was really exciting. It's like, wait, there can be entanglement in the warm, wet world up here, maybe. Questions, please.
I have a question about the discussion on dehydration.
Okay.
And the data of diffusion was showing basically faster diffusion at high temperature. Is that right?
Yes.
So I'm wondering if there was any convection effect. You know, you are using something like 70 degree, right?
Yes.
So the convection effect in diffusion experiment can be quite significant. Have you assessed such convection effect?
Oh, convection. We're actually, so yeah, I think the ThOC is performed without the convection interaction at a higher at elevated temperature. So not our case. Yeah.
Thank you.
Other questions? Well, I'm just curious, maybe the initial motivation or curiosity about Posner complexes, but do I understand you correctly that this depletion interaction, which is at all compartments in the cell, you think this could be a much wider spread phenomenon inside cells? Is this something you think related to these droplets that people are all excited about with these macromolecules coming together in these droplets, these reversible membraneless organelles? Do you think these kinds of phenomena might be playing a role in that?
I'm sorry.
So I don't know if you think that this is a general phenomenon in cells and related to the membraneless organelles, the droplets that are now thought to be biologically relevant.
Yes, absolutely. This is a common phenomenon we observed in a wide range of phosphate-containing species, also some biological molecules in vivo. So yeah, that's why we suggest that when you, for example, when you study their role, for example, in the biocatalysis or the formation of some biomaterials, you have to consider such assemblies when you interpret your data, both in vivo or in vitro.
It seems like this could be something in-cell NMR might be interested in finding some way to track this down inside living cells.
Yeah, sure.
Okay. Oh, good question.
I have a much more basic and a naive question. So all of us use PBS in our NMR. Now, should I use it to study temperature dependence, or would I have a different property when I go to 50 degrees? Is that a problem, or?
No.
So it's not a problem, we can still use PBS? So my question is that, let's say if you're using PBS as a solvent, as a buffer, and then you're looking at temperature dependence, should I be worried about this phosphate aggregation and or whatever that form that it forms? And
yes, absolutely. Actually, I can let you know based on our simulation results that this aggregation population is small. Like it's like 0.13% at room temperature and 0.73% at high temperature, like 70 or 80 degrees Celsius. But even though it's small, it does have significant impact on the biological assemblies between the phosphate-containing species in closer proximity. So yeah.
And have you looked at the same in a serum condition, which is what will happen inside our bodies? Have you, instead of looking at a buffer, have you looked at it in serum or something that mimics what would happen inside the body? With this, is the same effect observed there as well?
Oh yes, it's the same thing. Yeah.
Okay, well, thanks for visiting ThOC, and thank you again. I was going to ask you if there was a switch. I didn't know about it. No problem. Okay. Our next presentation will be by Alvar Gassert from the ETH in Zurich. And it's a nice follow-up, I think, to, or not necessarily follow-up, a nice complement to the first talk.
Okay. Yeah. Well, I also want to start off with thanking the organizers for giving me the opportunity to present our work here. My voice shouldn't fail right now already. Okay. Now, also thanks to Harry for introducing fluorine. So we're going to go on with fluorine. And Harry introduced that fluorine has this huge CSA, right? And what we're going to do is we're going to take it to our advantage. We're going to profit from the extremely fast relaxation that it has. And we're also going to profit from the fact that we can calculate the CSA. And it's actually also thanks to Ilya, thanks to him, thanks to a quick email exchange with him that got us started on calculations of CSA. But let's take a quick step back. What does our lab do? We're interested in understanding the molecular mechanisms of human disease. And therefore, we spent most of our time working on those difficult big human proteins, membrane proteins, GPCRs. And we need to develop a lot of methods to be able to work with these. And that's what I wanted to show here. So go check out the posters of Arnel and Philip and Simon if you're interested in this. Now, Simon is also the most important person on this project. That's Simon here, filling the magnet. Both of us used to work at Novartis in drug discovery, and he moved with me to ETH. And our second important part of projects at ETH is trying to find new starting points for drugs. And this is what this talk is going to be about. Now, let me quickly go through a typical drug discovery workflow at the very beginning of a drug discovery project. NMR is a very well-established technique in doing this. So we screen hundreds to thousands of compounds. At Novartis, we used to screen 4,000 fluorinated compounds in a library. From this, you get hits, so you get ligands to the protein. And these can be between 20 and 40, maybe. And now comes the big work, because this part now is really automated. It's very well set up. What you now need to do is to figure out which of these ligands are the strong ones, which ones deserve being worked up and put chemistry on. Because our screening experiments, they are quick and dirty. They just give us a yes-no answer whether a ligand binds or not, but they don't give us an answer whether a ligand binds strongly or weakly. And for this, we need to get the affinity of the ligand. And this is usually done, or actually the only good way to do this is to do protein-observed titration experiments. You need isotope labeled protein, which is very difficult sometimes to get for human protein targets. And what I want to talk about today is first, how we built a screening library for fluorine screening in academia. And then we actually came up with a new way of doing very efficient ranking of the affinities of these ligands. Now, I need to give you the very first principle because it's going to be important later on how we detect ligand binding. Probably most of you know this. Let's look at this ligand. It's a ligand which contains a fluorine group, which we're going to see now in the fluorinated spectrum. If the ligand is free, it's tumbling very quickly, very fast in solution. We get a sharp line. if this ligand is bound to the protein, its ThOC is slowed down. So it's on a very big particle, and we get a very broad line. So we profit essentially from this extreme CSA effect because we have a huge contrast between unbound and bound in the spectrum. What actually happens with those weak ligands is that they constantly exchange between bound and unbound, and we will also need to worry about this exchange. Just another way of looking at this, so this is the signal of the free compound here in gray, so it decays with the rate of half per second or so. Now, if it's attached to a protein, the T2 becomes very, very short, so the signal essentially just disappears. Now, since we're constantly switching between free and bound state, we get some average. It's all in fast exchange, so we get this nice average relaxation behavior. Now, the relaxation in the bound state is often more than 100-fold stronger than in the free state, such that we can detect binding even if the ligand is less than 1% in the bound state. So it's a very sensitive method. So that's one of the powers of NMR, that we are extremely sensitive to binding effects. That relies in this. What we do, instead of taking entire relaxation curves, which would take some time, we want high throughput, right? We just take a spectrum at one point, here maybe at 300 milliseconds relaxation delay, and then we just read out the signal intensity. And then you nicely can see a contrast between free and bound. Okay, obviously, this can be easily described. The blue line, essentially, this relaxation, is composed of a weighted average between the relaxation in the free state and the population weighted relaxation in the bound state. And the affinity, how strong a ligand binds, is encoded here in the, what's the population of the bound state? A strong ligand will spend most of its time bound to the protein, and the weak one will only visit it occasionally, right? And then something which complicates things is the exchange, which will have an additional relaxation effect, but we'll come to this later. But with this, we have covered all the theory, and we're set to do experiments. Now, this is where we get our sensitivity to binding effects from, and now we can go and do screening. We don't have much of throughput yet, so these experiments take maybe 5 to 10 minutes each. If we want to screen 1,000 ligands, that will take a while. And of course, this has been done for decades. What we do is we work in mixtures, and also here, fluorine has a huge advantage. If you look at this representative small molecule, it has about 10 hydrogen atoms, and therefore each molecule gives us 10 signals. So we very quickly run into overlap. And therefore, we can make mixtures of about 8 compounds. Now let's look at the mixture of fluorinated compounds. Typically, each compound carries just 1 fluorine. And you see here a mixture of 45 compounds at the same time, and there is less overlap than in the upper spectrum. So we can make much mixture of many more compounds, and therefore we get much higher throughput. With this obviously comes a problem. If I draw the hydrogen spectrum to scale, what you see is that we have a huge chemical shift dispersion in fluorine. That's great for overlap, but it's a problem for NMR. We cannot excite this entire spectrum. So essentially we need to chop it off and do different experiments because our pulses just don't cover the entire spectrum. Now, to solve this problem, we teamed up with Burkhard Louis in KIT, and he designed very nice optimal control pulses, which are capable of covering the full 200 ppm that we have in our fluorine spectra. And we can do now all our experiments. We have 90 and 180-degree pulses, which have the full width. So over here, you see the essentially ideal profile of these pulses. So now we've worked everything out, and now we can go and screen. And we've published this. So this was the library we set up at Novartis, where we had nearly 4,000 compounds that we screened very efficiently. And then you just look, and the analysis is very simple. You just go and try to see where you have strong changes between the once you add the protein, right? Okay. Now, when we came to ETH, we saw we don't have a library. So the first step of what I want to talk about here about new things is how to build a screening library. And again, this is Simon. He's into chemoinformatics. So what you do is you essentially get all the ligands you can get from catalogs. You don't buy them yet, so just virtually. And then you put them through a series of filters. So first you want to only have the small ones, which are fragment sized. Then you want only those which have a fluorine. Then you go and try to have an idea of the solubility. Then you try to have a large diversity in your ligands. You want to cover all possible chemical space. So if you get clusters of similarly looking ligands, you just want to have one that you are going to screen instead of screening 10 very similar compounds, and so on and so on. And with this, you end up with a certain number that you predetermined of compounds. And in this case, we had about 1,400 compounds that you would then actually buy. You look at different dimensions of your library like this, and then you massage it a little bit so that you come up with what you really want, very nice diversity and a good coverage of chemical space, as it's said in the drug discovery jargon. Okay, now we buy these compounds, and now comes the hard work. We really need to test them. Are they soluble enough? Are they pure? You don't want too many contaminations in your compounds. And also, don't they relax by themselves? There are many compounds which just tend to aggregate, and they will give us the same readout as if they were binding to a protein. So we don't want these compounds. And really what turns out to be the issue is solubility. So nearly half of the compounds don't meet our solubility criteria. And so imagine you need to measure 1,400 compounds. You need logistics. So we have every compound in a 2D barcoded tube. We have dozens of those racks in our freezer. We have a database. We can scan them. We always know exactly what's the, we know what's where each compound is stored and so on. And then we have some robotics to fill the tubes. And then obviously, I'm not going to make a Bruker advertisement here, but we then have the high throughput sample changer to be able to look at all of this. All right. So we now have this library, which 800 fragments passed our quality control. We screened them in mixtures of 24. We have already screened them on quite a number of targets. Here is just one example, the COVID main protease. And for all of these targets, we got hits, and here are the hits that we got for the protease. One thing here that I have an audience, we plan to enlarge this library. I have some money to enlarge it, but if you also are thinking of doing a fluorine library, we should combine. We should try to do a crowdsourced library that we can all profit just from having more compounds that we can screen. So from the NMR perspective, we can screen 4,000 compounds in 24 hours. And that would be a very nice place to go. So I'm just telling everyone who's interested, and maybe we can combine. Okay. Now, this is it. We have our screening library. We can screen them. We have our initial hits. And now what we need to do is to figure out which of these hits in here is the one which, or are the ones which we should first work on and develop further. Sorry. That summarizes essentially what I just said. I forgot I need to say. Now we go to rank our hits. So I show here two representative ligands of trypsin, actually it is. And you see that this ligand in the fluorine spectrum produced a very strong relaxation effect when in presence of the protein, and this didn't produce such a strong one. You would immediately say, well, this seems to be the stronger binder, but it's not the case. So actually this one is 10-fold stronger than this one. And this relaxation business, it's just complicated. You have exchange contribution, you have different CSAs for these different fluorines, And obviously, you also have dipolar relaxation, which is going to be different. And what we want to do is actually to figure out whether we can improve that. Now, the standard methods of getting KDs are either protein observed or ligand observed titrations. And as I said, protein observed titrations are the gold standard. However, now you need to go to the lab, you need to produce your protein if it works, and you will need quite some amounts of proteins. So why don't we do ligand observed as we did our first screening experiment? The ligand observed titrations work like this, that you essentially always record a relaxation curve at different protein concentrations. So we do this here. We go to fairly large protein concentrations for drug discovery experiments, like 80 or 100 micromolar. And we get these curves, and in principle, it looks nice, right? So what's wrong with this? This is rubbish. So this is not going to give you a KD at all. So the problem is we're not sampling enough. I'm just drawing this in the lower corner here. This is what I showed you before. What one needs to do, obviously, is to follow the entire curve until it plateaus. So if you have a KD of 250 micromolar, it's going to plateau at about 1 millimolar protein concentration. Most proteins that we work with, we cannot concentrate to 1 millimolar, let alone produce these amounts. And this curve fits these points that we recorded very nicely, but so do all of these curves. And they range 2 orders of magnitude in KD. They all fit these points. So how could we get a KD or an affinity from a ligand observed titration, which is so easy to do? Well, the plateau is essentially it's the relaxation in the bound state. So if we are able to calculate the relaxation in the bound state, then we know where the plateau ends, and then we can fit the curve. And this was our aim in this project. Now let's quickly look at what seems a bit complicated or a bit crowded. So you know that the relaxation we measure is composed of these components, and we are now interested in whether we can get the bound state relaxation. Now, this is composed of CSA relaxation and dipolar relaxation contributions. The CSA depends on the shielding tensor, which we can calculate in DFT calculations with quantum mechanical calculations. Dipolar relaxation, we will never be able to calculate. We don't know anything about the distances between the ligand and the protein in the bound state. So this is not going to work. However, if we go to high fields, you see here the CSA relaxation depends in the square to the B0 field. We can make this much, much bigger so that the small differences in dipolar relaxation are not relevant anymore. And this is what we do. So we actually can measure the free state relaxation, we can calculate the bound state relaxation, and we measure at high field such that we can neglect the dipolar relaxation. And high field is like 700 megahertz. And we still have this exchange issue here. And this we just suppress with NMR techniques. So what we do is we do an on-resonance spin lock. And thanks to Bruker cryoprobes, we can do this at 10 kilohertz. And you see that's more than what we need. So here we have strong exchange and we increase the spin lock, but we really get rid of the exchange contribution. And we tested this with many compounds. And now I need to speed up. We calculate all the CSAs for the fluorinated fragments. And then we did our ranking for the different proteins here. And we also checked against established methods like reporter assays to get to figure out whether we have the correct ranking, and we get this very nicely. And then finally, this is what we get. So we get a ranking where I get you the gold medal, the silver medal, and the bronze medal of the different compounds we found. And this is what we want. So we prioritize these compounds over others. I even put here the KD in micromolar. We typically just then determine one affinity, And because we have the ranking, it's proportional. We have the relative affinity of all the others. So then we can get numbers to it as well. All right, that's the summary. So we have a library for which Simon now has calculated for each fragment before upfront the CSA, so that we can just take it. And we can then go into ranking using the CSAR method that I have shown you to you now. Okay, so I would like to first thank you for your attention, and then Simon, who was probably invaluable for this project, Gabriela, Niels, Philip, and Daniel, who are over here, and Oli for help on the DFT, and Helena helped us with the spin-lock experiments. Okay, thank you very much. Sorry for being a bit over time.
Thanks, Alvaro. So we have time for maybe a question. Okay, I have a question. Is does it? I assume this does not have a major effect since you showed the rank ordering, but does one have to worry about the CSA properties and for a bound ligand differing from the free?
Yeah, very good question, and we checked this. So probably the electron density or so will slightly be changed by binding effects. And we did things like, for example, protonated, deprotonated state of the same molecule. Actually, it doesn't make such a big difference. So these fluorine tensors, they are quite robust. So we see differences in the order of 10%. And that's on a ranking that doesn't make a big deal.
Maybe based on what earlier, if I had a covalent binder though, that might be a very different story.
That might be a different story, yes. But that's, we anyway not going to be able to do anything about this.
Oh, go ahead.
Not only a covalent binder, if you have a very tight binder, you probably will also not see it, right? If you have an nanomolar binder, you'll not see it as well. I have two questions. Do you need to worry about proton decoupling? Because some of these fluorine proton couplings could be strong. Yeah. And I think most of your fragments have only one fluorine, except for the CF3, which is equivalent. Because you're doing a spin lock, would you need to worry about the ThOC effects if there are more than one fluorine?
Okay. So typically we have either CF or CF3 moieties. Here we would not need to decouple, but here we need to decouple protons, absolutely. So it's like 10 times more signal because you get these complex coupling patterns. We actually expected from the decoupling to have additional effects like STD or so, but there's very little happening there. And we take care not to have coupled fluorines, because that makes things messy, as you say.
Okay, if no further questions, let's thank Alvaro again. Okay, and our final presentation will be by Lorraine Brennan from UCD Dublin, and metabolomics and nutrition research. Whenever you're ready.
Okay, thank you very much to the organizers for inviting me here today, and I'm delighted to be in person, enhancing my vitamin D content in a bright and sunny Florida. So today, what I want to do is to cover what we're doing in the context of metabolomics. So I'll start off by giving an introduction to metabolomics in the context of human nutrition, and then I will talk specifically on an area that we're particularly interested in, and that is biomarkers that tell us about what we eat. So just to quickly summarize metabolomics, of course, we're measuring small molecules, we're measuring them in biological samples, and we work predominantly with humans, so we're trying to look at urine samples and blood samples predominantly. And we measure these molecules, these small molecules in NMR, for example, but actually we also have to acknowledge that we use a lot of mass spec as well. So my lab will use both NMR and mass spec and try to combine them where possible. So what I'm going to talk about today is actually work that we have done with NMR and using NMR only. So if I take a broad look at what metabolomics does for human nutrition, you can broadly categorize the applications into these 4 areas. The first one is biomarkers of food intake, and that's what I'm going to talk about today. The second one is the area of precision nutrition, where we're really trying to move away from one size fits all, and we're trying to give more targeted or more guided dietary advice to individuals. The third is trying to understand the molecular understandings of the impact of food on health. And the third is the study of diet-related diseases. So the first one is biomarkers related to food intake. And you might say, well, why are we interested? Why are others as well? Well, it's actually a really big issue in nutrition research. So the cornerstone of many nutrition studies is being able to understand what people eat. And our current methods are extremely lacking. So currently we rely on self-reported data. That is, we ask individuals what they're eating, and this has well-established limitations associated with it. So our current approaches include food diaries, food frequency questionnaires, and 24-hour recalls. And food diaries, for example, you catalog the foods as you're eating them. Food frequency questionnaires, you have to remember what the frequency of a list of foods that you've eaten over the last year, for example. And then 24-hour recalls, you have to record or remember what you've eaten in the last 24 hours. So not surprisingly, these have limitations associated with them, such as people will over-report foods that they perceive to be healthy and will under-report foods that they perceive to be unhealthy. So there's a real need for more objective measures of intake, and this is where biomarkers come into play, and in particular, these biomarkers related to food intake. So the concept behind these biomarkers is that they will help us increase the accuracy at which we assess people's dietary intake. And there are many examples in the literature of papers where people have used metabolomics approaches to identify biomarkers of intake. Now, I'm not going to talk about the discovery of these or to go through any of them. I'm going to focus more on how we could potentially use these biomarkers of intake. So how can we use them then? And again, I'm focusing in on the context of human nutrition. Over here on the left is the first example where we're just simply using them as a measure of adherence to a dietary intervention. And this is really important for those of us who work in human nutrition, because this can help enhance the authenticity of our trials. Measuring of objective measures of intake, that's what I'm going to talk about today, and how can we use them to correct self-reported data. And then finally, we can use them to look at the relationships between health parameters and disease. So ultimately, if we're here in this area looking for objective biomarkers of intake, what we'd really like to do is to take a urine sample, determine a biomarker concentration in that urine sample, and tell exactly what an individual has eaten in terms of grams per day. So we set out to see, well, actually, could we do this? And could we do this with some of the well-established biomarkers that are known in the literature? So we set out with this, as I said, we performed a targeted metabolomic analysis looking at a specific biomarker called proline betaine. And this had been well validated in the literature before we worked on it as a biomarker of citrus intake. We performed a very detailed intervention study where we were looking at where we asked individuals to consume certain amounts of citrus, and we measured the urinary excretion of proline betaine. And our hypothesis was that as the portion size of citrus increased, the excretion of proline betaine would increase. And you can see that that happened very nicely here. But the important aspect of this calibration curve then is that you could take any urine sample, and then you could plug it into this equation, and you could determine the intake in grams per day. So as I'm at an NMR conference, I decided I'd just put up quickly a quick snapshot of a human urine sample. This is just a very small part of the region. And we're particularly interested for this study on proline betaine, which you can see over towards your right. So our hypothesis was, or what we started out to do, was to try use this biomarker measured in urine to tell us about the exact quantities that individuals were consuming in terms of citrus. So this is what we call our biomarker data, or our biomarker approach, and we get an estimate of intake from that. But we wanted to compare it to the gold standard method of measuring food intake, so we compared it to what we would get from semi-weighed food diaries. So we had 4-day semi-weighed food diaries as well, and we could estimate from these individuals' habitual intake. So to do this, we looked at a nationally added representative sample of the Irish population, so 600 samples. We took their food diary data, we pulled out their citrus consumption, and we have a reported citrus intake. We took their urine samples, we measured proline betaine using NMR, and we plugged those proline betaines into the calibration curves to estimate our proline betaine intake. So we had a predicted citrus intake and a reported citrus intake. But before I look at that in the observational study, I want to go back to our intervention study, because in our intervention study, we had large numbers, so we were able to develop those calibration curves using 2/3 of the data, and then using 1/3 of the data to compare with the, to really test out, could the urine sample or biomarker data actually predict intake and compare it with actual intake. So we done that, and you can see a snapshot of the data here. So on the right, you have what's orange juice predicted using the biomarker, and then to its left, you have the actual intake of orange juice. And you can see that the biomarker worked really well across a range of intakes. So down at around 50 grams per day, it worked well, and right up to 250 grams, it worked well. If we do a simple correlation analysis on the predicted orange juice intake from fasting urine sample and actual orange juice intake, we get really high correlations of 0.9, and if we use a 24-hour sample, we get similar results as well. So this was very exciting for us, that we could see in our very controlled intervention that the biomarker could really predict intake of citrus. So then what happened in our observational study in our larger population group, if you remember back, we wanted to take urine samples, plug them into our calibration curves, and then predict using urine biomarker data only what amount of citrus individuals consumed. And again, you can see a snapshot of the data here. On your right is the predicted citrus intake, and on the left of that then are the recorded or self-reported citrus intake. And again, across a range of intakes, we got relatively good agreement between both methods. We compared both methods across the population group using a Bland and Altman plot. So that's what you see here. So what you can see here is that the mean difference between both groups was very small, but there are individuals where there was big disagreement, and you can see that up on the top right. And if you remember back, when I started off, I said that we have limitations with our self-reported data. So we would expect there not to be complete agreement between the two methods. And in fact, what's happening here, these individuals here are underreporting their intake. So we're getting a truer reflection on their intake using the biomarker data. So this was very exciting. We were now able to say that we could use these biomarkers to help us understand what individuals, and I'll remind you, this is only one food, and we eat much more than one food, but still very exciting milestone for us. So we wanted to see, well then, what could we do with this type of data in large population groups, and moving it into epidemiology? Could we use these biomarkers to correct measurement error in large population studies? So what we done here was we took the biomarker data and the self-reported data, and we modeled these two data together. We took the estimates from that model, and we used those estimates to correct our self-reported data. Now, this has advantages in that if you're going into large population sizes, it means you don't have to have biomarker data for all of the group. So we tried this out again in the Nationally Adult Nutrition Survey in Ireland. So we had 800 individuals, and we wanted to see if we could take this approach. So we developed these calibration equations, where M here is the biomarker derived estimate of citrus intake, and W is the mean daily self-reported data. We model these two together, we get the estimates from them, and we can then derive a calibrated mean daily citrus intake when you only have self-reported data. So the concept behind this is that you're correcting for the measurement error, or some of the measurement error in the citrus, in the self-reported data. So we done this on our 800 individuals, for which we had only biomarker data for. We used only biomarker data, but we had self-reported as well. And you can see here that it worked pretty well. So we were able to predict their intake. We developed an app associated with this and an OR package, so it's all freely available for people to browse through and to have a look at it. So then the next question was, well, that this works really nice. It worked for us in our population group, but in terms of epidemiology studies, 1,000 individuals is relatively small. So we asked the question, well, how much biomarker data do you need? So we done some simulation studies where we're trying to estimate how much biomarker from a population group you would need. And in this context, we found that you needed about 20 to 30% of the population with biomarker data, because around that, you got stabilization of the standard deviations. That simply means that any extra biomarker data wasn't really improving the estimates of the calibration equations. So just to summarize that part then, so this is a very nice example of how we use self-reported data, both in combination with the biomarker data. We model them and develop calibration equations, and we use those estimates then to correct the larger sample size. And this has really great potential applications when we move into large samples and when we move into studying epidemiology. Now, Oz and others have noticed as we move through this field and we get more and more information about biomarkers of intake, that actually it's rarely going to be one biomarker, one food. What we've seen and others have seen is that it's actually panels of biomarkers that are more representative of a particular food. So we were interested in looking at this concept of panels of biomarkers. So again, we performed a dose response study where we had individuals over 3 weeks consume different portions of fruit, and we collected urine samples over the course of 3 weeks. And then we had a cross-sectional study again, where we had self-reported data and we had urine samples. So again, using NMR, we measured a panel of biomarkers, and using our dose response study, what we came up with was biomarker cutoffs for that panel combined that could categorize individuals into different categories of fruit intake. So we have 0 to 100 grams a day, 100 to 160 grams per day, and greater than 160 grams per day. And this was done, as I said, in our controlled intervention in our dose response study. So then we checked it out and went to see, well, actually, does it work in our free living population when we're not controlling everything about the individuals? And when we look at that in our free living data, this is on the right is what we estimate categorized individuals into different categories using the biomarker data. And in the middle, we have our self-reported data. So the biomarker data and the self-reported data agree very well in the kind of medium intake of fruit. We estimate using the biomarker data that people consume much less fruit than what they self-report. And again, up at the higher levels, we estimate that much less people consume higher amounts of fruit. And again, we would expect that difference between the lower levels and the higher levels based on what we know about self-reported data. So others are also interested in these panels of biomarkers. And here is just one example I took from the literature from Claudine Monac's group, where they were looking at banana intake. And they used a combination, again, in a multivariate model of biomarkers to classify individuals into consumers and non-consumers of banana. And you can see that up here on the ROC curve on top, that they had very good ability of classifying individuals into consumers and non-consumers. So working with these panels of biomarkers opens up lots of different opportunities for us, but it's actually also quite difficult. And it's difficult from a statistical point of view. So if you remember back on the left-hand side there, or sorry, on the left-hand side, I have shown the proline betaine example against orange intake. But what happens then if you have multiple biomarkers against a continuous intake of a food? So to address this, we developed a new framework called a multi-marker framework, where a new statistical approach of trying to model multiple biomarkers against a continuous intake of food. Again, we made an R package and a Shiny app, so people can use this and it's freely available. But in essence, what happens is that we have the multiple biomarkers. We model these against the continuous intake of food. We get the model estimation from that. So therefore, you can come along with your biomarker data and plug it into the model estimation and get back your predicted intake. And what's nice about this is we don't just give you an estimate of intake, but we actually also give you a confidence interval around that. So again, I said this is available, so it's free to use, so please have a look at it. And then for the last few minutes, I'm going to talk about dietary patterns, because you know that we don't eat foods in isolation, we eat them together. So from an epidemiology point of view and a health promotion point of view, we're actually interested in that combination of foods. So how can, and we asked a question then, could we use metabolomic data and biomarker data to classify individuals into different dietary patterns? So a number of years ago, we done a very crude model where we developed a multivariate model to classify individuals based on their urinary biomarkers into a healthy dietary pattern or an unhealthy dietary pattern. And more recently, we have sophisticated that model a little bit more, and we have defined 4 dietary patterns solely based on your urinary metabolomic profile. And now we've developed a model where you can classify an individual into one of those 4 dietary patterns. We then asked, well, could we reproduce this in a different population group? And then was it reproducible over time? Because we do know that individuals tend to follow their same dietary pattern. So we would expect that it is reproducible over time. So we identified these 4 dietary patterns. So we have a moderately unhealthy dietary pattern, a moderately healthy, and then a healthy or a prudent dietary pattern. On the top 2 scores here, you can see these are scores that we use as an overall indication of the diet quality. And you can see that these are higher in the healthy dietary pattern and lower in the unhealthy dietary pattern. And we have a whole range of different foods and food items that are significantly different across these different dietary patterns. I should emphasize that these dietary patterns were solely based on, our classification into these dietary patterns was solely based on the metabolomic data. And here we're using now the dietary data to see, does it make sense what we ended up with from the metabolomic data. We had blood samples and data on these individuals' nutrient status. So again, we looked across this at the 4 dietary patterns, and as expected, we have a very different profile of their vitamins across the 4 dietary patterns. So for example, serum folate is higher in the cluster that was deemed as the prudent intake, and whereas urinary sodium down here was the highest in the unhealthy dietary pattern and the lowest in the healthy dietary pattern. So what we had here then was a multivariate model based on metabolomic data that could classify individuals into 4 different dietary patterns. And then we wanted to see, could we reproduce this in an independent study? And then if it was reproduced, then how reproducible was an individual over time? So we did reproduce it in an independent study of 160 individuals. And here you can see the moderately unhealthy dietary pattern, and over on your right-hand side, you have the prudent dietary pattern, sorry, the moderately healthy and in the prudent dietary pattern. And what you're seeing here are 4 different visits. So these are 4 different time points for individuals, and you can see that the dietary patterns are being tracked over time. So we've got a very stable model that's able to classify individuals into a dietary pattern, and that seems to be stable over time. So then finally, if we think about these biomarkers, what I've talked about today is biomarkers that are really related to food intake. But not all biomarkers are going to be like that. They're going to be influenced by our microbiome. They're going to be influenced by our genetics. So what we're moving towards now is to try to develop models for bringing in that extra information and modeling that impact of food intake, the microbiome and the genetics on the biomarker to allow us then to look at relationships between food intake, disease risk factors and health parameters. And this is an area that really I think is going to take off in the next few years and has the potential of giving us very, I guess, sound information or accurate information on the relationship between our food intake and disease risk factors. So then just to conclude, I hope I've shown you that metabolomics, and I've spoken about metabolomics in the context of NMR-based metabolomics today, can give us information about food intake biomarkers. I've spoken about how we can use those food intake biomarkers to determine intake in individuals. The example I gave you showed that they compared very well to 4-day food records. It has the potential for classification of people into different categories of fruit intake, and that was the example using panels of biomarkers. And then the combination then of using biomarkers with self-reported data has huge consequences and advantages for use in the field of epidemiology. There are, of course, lots of challenges in this area, and I'm just going to mention a few. Unassigned features is a big issue, both in LCMS data, but actually also in NMR data. in a lot of the features that we're interested in, particularly when we look at mass spec-based approaches, are not in our classical databases. So we have to spend a lot of time trying to identify those features. And there is really a need for an international alignment and efforts in this space. NMR can play a key role in helping this assignment, but it's really underutilized, I think, in the field of general field of metabolomics to help these unassigned features. And combining NMR and LCMS, it really, again, is an area that we're starting to look into and to try push forward. So finally, I'd like to thank the wonderful group that work with me in UCD, thank all my collaborators and funders for supporting our work, and thank you for listening.
Thanks very much. Jim.
Yeah, I was curious about the proline betaine. It showed very nice correlations with food intake. And I would think most markers that you pick out would be sensitive to other variables like exercise and so forth. Is there something special about proline betaine?
So proline betaine is actually found in the citrus fruit itself. And a majority of it, when we ingest it, is actually excreted in the urine. We looked at if there were differences in that excretion pattern across sex, and we didn't find huge differences. So in that particular case, once you use fasting samples and you correct for osmolality of the urine sample, it works well. But you do need to correct for the osmolality of the sample.
It's very exciting to see nutrition work using NMR. I'll just follow up that question. I was also wondering, I know metabolites probably is highly regulated in blood, so it should be like stable with like, but in urine, I'm wondering, like, do you have to correct for like daily water consumption? Because like, I'm just thinking maybe you have to dilute, then is that going to affect any of your correlation with your intakes?
Yeah. So all the data that I showed here is corrected for osmolality. So that would take into account water consumption from individuals. Yeah.
Thanks.
And it's really important to do that to get those high correlations. And then if there are other factors that influence it, we can introduce them into our models. For example, our calibration curves. And once we have those in as covariates, then and once we know that covariate for the individual, then we can also take that into account.
Other questions? Well, this could be very useful for trying to address global health, certainly, to see how these profiles based on metabolomics can tell us about how disease might be more likely in one region than another. You said something that was, it implied people were dishonest about what they were eating versus what the data showed. And that brought to mind, if you do these studies like geared towards age, so you take the group from age 2 to 10, they've got to be off the charts or something, right? It's just mac and cheese and all this kind of stuff. I mean, is that something you notice, that the age profiles for these readouts show very different distributions according to the betaine assay, for example?
Yeah, so if we were doing a study, we would try to look at age. So we try to bring in age into it and then see in the larger population group, is age having an impact on the concentration of that metabolite in the urine? And if it is, then we have to be really careful then in our calibration curves that we include age as a covariate in that, and that it's modeled into that.
Okay, I mean, I was thinking you would use it as a diagnostic, basically, to say, look, I don't care if you want to eat this junk. I mean, you're just off. You're in a very unhealthy regime according to these profiles, right? So that's
yeah. You could do that. So you could just say, well, actually, whatever you're eating, it's just really bad. You need to improve your diet. Yeah.
Okay. If there are no other questions, let's thank Lorraine again, and in fact, all the speakers for this session.